All right, everybody. Welcome back to the Longevity Acceleration podcast. Today we have Peter Fedichev, who's got a background in physics and has shifted into the field of biology, which is something that the LBF is particularly excited about because we like to bring talent from other industries into the longevity biotech field. I think Peter's story is going to be inspirational for a lot of people that might be able to follow in his footsteps. We also have Aleksandr Sviridov, who I think is responsible for the Gero app, which is part of the two prongs of Gero. One is the therapeutic development and the other is the app. I think I'll let you guys introduce yourselves and explain more precisely what you guys do and how you got into longevity.
Peter,
Go
I'll
ahead.
let you
Okay,
start.
my name is Peter Fedichev. I graduated from Moscow Institute of Physics and Technology at Soviet MIT. Got my PhD from University of Amsterdam. I had a decent job. I was doing theory of correlated systems, condensed matter physics of strongly interacting systems. Then somebody showed me animals that do not age, and I'm happy that I switched. I think living systems are complex enough, correlated enough. There is a lot of physics toolbox that can be applied to understand negligible senescence. And we founded a company backed by adventurous inventors with the idea to make humans non-aging species eventually.
So I'm Aleksandr. My last name is Sviridov. I actually have a background in computer science, and before joining longevity field, I was doing mostly. I used to be a founding engineer for many mobile app website slash website based startups. Some of them quite successfully. Some were acquired, including by Samsung. So roughly in year 2019, I realized that I'm probably having a sort of an existential, not really understanding what is it. I guess I lost motivation in doing things, and I was trying to understand why is it the case. And I started to look for new opportunities and for new meaningful projects. And health and longevity in general was one of the directions which I outlined for myself. And eventually, with several years of trials and errors, I learned that I need to focus on longevity myself, because previously I thought that maybe I can just earn a lot of money and buy products developed by incredible smart people who understands biotech way better than me. And two things that I learned that changed my mind is that it's not that many people actually working in our field, and I can do a contribution that will have an impact. And the second thing which I learned is that I actually cannot think about anything else. So I have to focus on longevity because I cannot do anything else properly. So that was the switch for me. It was a bumpy road. I initially joined a community called Open Longevity, which is the biggest Russian speaking community around longevity topic. And together we launched several initiatives. I helped them to launch networking initiatives where one of them called Random Coffee. I met Nathan and Peter for the first time through Random Coffee. But since I have a background in computer sciences, I was always looking for opportunities to apply my skills in longevity field, which turns out to be quite hard. It turns out if you're not an ML engineer or bioinformatician, it's not a huge demand for your skills in longevity field. But I was lucky to meet Peter, Maxim, Alexei, and other people from Gero offline in Zuzelu, and we had a long and which resulted in me joining Gero and leading development of GeroSense, including the latest initiative called GeroSense Studies, and also some of the compute-related, cybersecurity-related efforts in Gero.
Yeah, thanks for the detailed intros. And we're also big fans of Open Longevity and collaborate with them quite a bit. The Save Forever campaign, I think, is a really cool ongoing initiative that has a life of its own now. in terms of bringing new people into the field. They've done a lot of good work, and I think we do the same thing. We're more on the European and North American side as they cover the rest of the world. But yeah, let's get right into the science now. First of all, I really like, Peter, the way you've brought new terminology and maybe precision in the way of describing aging that previously didn't exist. So the separation of entropic damage versus dynamic instability. I think quantitative ways of thinking about aging and modeling aging are necessary and important versus the sort of, in the past, we've had a lot of reductionist narrative based theories of aging, which is a different way that probably is a result of your physics background. So I was wondering if you could describe your model of what you think aging is and how it can be broken down into different parts.
Yes, maybe first I will comment about the cultural difference of narrative and qualitative and semi-quantitative sciences, maybe. I'm actually entertaining ourselves with the idea to write it down, because when you come from physics, they teach you a certain way how to build models and what is called the theory. And then other sciences also use the same titles like theories. And that's how you can wake up in a world where there are 500 theories of aging, which is a bit strange because in physics we still have a standard model that looks like a theory because it doesn't explain only experiments with an error 1/1,000,000, but it's still not called a theory because we know that it is incomplete. So if you look how physics was coping with complexity over the last almost 100 years, I think you can find an interesting pattern. So whenever something complex was identified experimentally, what physicists tried is first to establish a set facts, a set of experimental truth, and then build a mathematical model that explains them with all these facts without explaining the phenomena. And there are multiple examples to them. For example, superconductivity, who found that certain materials do not have resistance. They have some funny properties regarding the magnetic field. Everything else, all of these measurements were combined into a set of facts, and then theoreticians explained it with a microscopic theory. So that's here I'm coming back to your idea reductionism versus this kind of emergent picture. So first people establish truth, then people try to build a model that makes comparison of theories with experiment simple and quantitative immediately. And then people try to build a microscopic theory, reductionist theory, that explains it all. So what I think, and that there are more examples to that. So for example, in cosmology, there is the standard model of cosmology that has dark matter, dark energy. We still don't know what is dark matter and dark energy, but we have all the numbers. All these numbers are being improved experimentally with new telescopes, with new machines. Experimentalists are improving numbers, and theoreticians are trying to improve the explanation of these numbers. So what I think is important here is that one theory is phenomenological, so it doesn't explain, but it describes the experiment, right? So phenomenology describes the experiment with numbers, but it doesn't explain why experiments happen in a certain way. And then there are microscopic theories that, of course, are friends of phenomenological theory, so they reproduce phenomenology, but they explain why phenomenology works in that or another way. So I'm sorry for being long on that, but I think what is maybe missing or should be done in aging is that first we have to agree on phenomenology. So for me, for example, it's very important that there are animals that age exponentially, like unfortunately we do. And there are animals that do not age exponentially or age exponentially, but is such a slow exponent that it cannot be measured, right? We have kind of two regimes apparently there. There are some subtle differences in behavior of these animals there as well. Mortality is just population quantity, right? It's not fun. Mortality doesn't exist for a single individual. It's a binary event. You're either dead or alive, right? So you cannot measure mortality in one. So somebody has to relate it to the character of changes, age-related changes that are measured on molecular and functional levels. So these things have to be described and agreed. And then there has to be a model that describes them mathematically without explaining, right? Because once we agree on this picture, experimentalists can try to improve the numbers, try to improve which animals belong to which category, for example, try to see if there are more categories of animals and everything like that. And once this is established, then every theoretician, like me, for example, could try and produce the microscopic theory, why? What should you be doing on a molecular level in order to produce this or that phenomenology? And as you can imagine, to produce naked mole rat phenomenology is a little bit more interesting than produce mice phenomenology, right? So it will happen, and it happens indeed, that there will be different microscopic theories. We know that different animals that almost do not age or do not age at all arrived to that situation in different ways on a molecular level. We have different examples of all kinds of rats, bats, the different species, that all of them got this interesting behavior doing different things. And this is remarkable because it means that they still do the same on the phenomenological level, and they're different microscopically, which means that there are many ways to achieve the same thing. So, like in superconductivity, you have at least two different kinds of superconducting materials with the same phenomenology. We have apparently many types of non-aging animals with the same phenomenology. And that's the way how you can still rigorously have many microscopic theories, right? Different people can be right, but just in different species. And what most important, that we have to have the same phenomenology. So back to your question, I believe that there are two kinds of aspects to aging. There are biological clocks, for example, right? There are aspects of aging that are reversible, and there are aspects of aging that are not reversible. As you may know, that there was a revolution in physics, that we know that in physics everything is microscopically reversible. You can fix everything if you want. But we also know that no matter how strict you are to your kid, in 1 year your Lego box cannot be assembled anymore, right? So certain things are just entropy. So there are reversible and irreversible aspects to aging. And there are animals where reversible aspects dominate, like mice, for example. So that's why there you can do rejuvenation, you can do interventions against aging pretty easy. And then there are humans where entropic component, the irreversible component to aging dominates, which means that the dynamic aspects are only important very late in life when people are almost dead. And that's why you have to have different aging clocks in humans. There are clocks that measure chronological age. This is related to entropy. There are clocks that are measuring time to death. These are related to dynamic aspects. And testing and optimizing drugs in mice, you get good drugs for mice, bad drugs to humans. And to find a good way how to find drugs that are reducing entropy, that are reducing the entropy production in humans. And with the last but not the least, you can revert everything microscopically, but it may be just too difficult in complex systems. That's why I believe, and that's what these theories are showing, and that's why we put another preprint a few weeks ago, suggesting that most interventions that have been studied in mice do not affect the reversible component of aging. We need to find species that look more like humans. We need to collect more data from humans, because we will be surprised how different things work against these dynamic and reversible processes. We need to be open to the fact that in order to change lifespan by a lot, we have to be doing things that do not help mice a lot. And that's why phenomenology is important, theory is important, and numbers are important. We are making the picture where both kinds of drugs can be tested. And eventually, we hope that one of them will lead to dramatic life extension in humans.
Awesome. Yes, that's a super good summary. And I think I want to do a little bit of translation here because you talk in terms that I think most biologists aren't as familiar with necessarily. And so sometimes I think there's a little bit of misunderstandings. So to try to translate, I think when you talk about the dynamic instability, this is usually what people would refer to as homeostasis, right? There's certain types of stresses and damages that are just part of the day-to-day process of being alive. And we have mechanisms to bring those back in line. And so that's what people would usually call homeostasis. And when you're young, it's fairly stable and robust. And as you age, it becomes less and less stable and robust. And at some point, the homeostasis fails and you go outside of range, and that's mortality. With entropic damage, those are types of damage that I think Aubrey de Grey would talk about as being the things that are accumulating that we don't have a mechanism to repair. So there is no homeostatic mechanism, but they accumulate slowly, right? And so they only start to have effects later in life because once they cross a certain threshold, they start interfering with your actual homeostatic mechanisms. And then that's when things start to go off of the rails. It's an exponential process because once one mechanism fails, there's cascading effects because everything has to work together. Biology. If one part of your biology is not working, then the other ones can't also because they all support each other. That's probably the biological sort of terminology.
Yeah. I like the way you put it. Yes, of course, in evolutionarily sophisticated animals like we are, most of the damage can be repaired. Not all, but most of the damage can be repaired. But it doesn't mean that we have time to repair it, right? So we have different priorities along our life. Certain errors, certain kinds of damage cannot be repaired or are not repaired, right? And this is an evolutionary decision what to spend energy for. If you live long enough, like we do, 50 to 100 years, even those things that are most often repaired, but sometimes are not repaired, they accumulate and they put stress on our all other responses like homeostatic equilibrium maintaining mechanisms. So that's why they are degrading linearly. And when your homeostasis maintenance mechanism degrade linearly, it can be easily shown that the probability of failure in these mechanisms is exponential. So that's how this slow build-up of technically reversible, but apparently not for technical reasons, builds up and produces exponential acceleration of mortality. The good thing in this picture is that this process is linear, right? So humans evolved. We're almost naked mole rats. We have just this small linear leakage that kills us in 100 years, which is pretty good, which means that strong life extension in humans should not be a very difficult problem.
Yeah, so I want to loop back later to the difference there of the approach of trying to slow down that rate of linear accumulation versus trying to actually create new homeostatic methods to prevent those accumulations at all, because those are different strategies. And that's, I think, a debate that was originally planned between you and Aubrey that hasn't happened yet, but I hope it does happen because I think it would be quite fun. But I want to loop back to something else that's a connection between, or a comparison between physics and biology, which is data. And I think historically, just the production of data in biology has been pretty haphazard and piecemeal and often not reproducible. The methods are problematic just because biology is so complicated and messy. And I think in the last 5 years, we've started to see technologies like single cell RNA sequencing, methylation, genome sequencing. And I think the new tools that we have in order to generate data are what is allowing biology to become a quantitative science. Whereas previously, I used to say that biology was a discipline for people that weren't good at quantitative thinking, but that might have been maybe too much of a hot take. I think it was more that we just actually didn't have tools to treat biology very quantitatively in the past, but now we do. And so that's a big change, which means that we should be treating it quantitatively. And I know you guys rely on methylation for the studies that you've looked at, but I'm curious as to other forms of data collection or data that you think might be important, because methylation is obviously this. If you have a hammer, everything's a nail. We have methylation and transcriptomics now. We don't have single cell proteomics or single cell metabolomics, and maybe those would be important too. So I just get your thoughts on what's the state of our ability to collect data in biology right now?
That's a great question because, interestingly enough, in some ways, we have had too much data in biology, but in a very bad way. In biology, most of the time, you measure very many features from very few experiments. And as you know, from statistical point of view, from machine learning point of view, this is a very bad idea. Because when you measure too many things, you have a lot of false positive signals, essentially, for everything. So in order to apply sophisticated methods like machine learning, you have to have what is called deep datasets, where you have samples much more than features. But biology is a very interesting area of knowledge where the total number of features that you can measure is just very large. Think about 1 million methylation features, for example. In order to have a deep dataset with single cell methylation, think about how many samples you have to produce to be deep with 1 million features. If you think about genetic features, even the total number of people living on Earth is not deep enough to really build foundational models with modern AI. So I think that's exactly where physics is very strong, because I like to bring up this example with thermodynamics. Thermodynamics as a science was developed to build a steam engine. Steam engine relies on molecules to work, but people could not measure molecules at that time. People were not even sure that molecules exist at that time. And still, physics allows you to build description, the same phenomenological description, with emergent quantities that do not exist on molecular level, like entropy, for example, right? In order to describe how complex systems work. So I still think that biology often ends up in this trap where you are always too shallow to work with your data, which means that you have to find ways to use statistical mechanics. I like to joke that what is called systems biology is called statistical mechanics in other fields. Statistical mechanics is a great way to reduce number of relevant variables to describe complexity. And then when you have this kind of reduced representation, you can actually use all that massive amounts of biological data that is coming. In biology, with all the possibilities. That's
It's
why
expensive.
we have to use this. It's not only expensive, it may end up being impossible in terms of the total number of people living, right? It's expensive first. It's much more expensive than with pictures. Remember that the revolution in machine learning came when the price of an image went to zero. In biology, the price of an image is still a few $100s, if not $1,000. So you cannot be as fast. This is still my position. We should be able to progress in biology even in situations when we don't measure everything, because eventually we will not be able to measure everything. Like you cannot measure the position and coordinates of all molecules in a steam engine, and you don't need to. So that may be an overcomplication. So now to methylation.
PV NRT, PV equals
yes,
NRT doesn't
don't
require
need,
you to know the position of every molecule.
Yeah. Boltzmann introduced entropy and shot himself because not all his peers were sure that this is interesting enough and molecules exist. This is a very bad story. And by the way, the reason why he was rejected by the community, because he was telling that entropy is going only one way, even though all microscopic equations are going back and forth in time equally well. And I'm just now enjoying that this discussion is coming, hopefully with my help, into the field of biology. And as a community, we have to progress all the stages of acceptance and anger against this phenomenon. So we have to relieve all these things. And in order to do that, we should drop this idea that let's sort these things out after we measure everything. First, it will take time to measure everything. And then even somewhere on the way, you will get lost with the total amount of things that you can measure. What I would try now to tell you that, yes, we looked at methylation. Why? Because if you want to write something conceptual in the field of aging right now, you have to show it in methylation, right? Because 5 years ago, everyone was happy with senescent cells. Now everyone is thinking about methylation. We always have this number one theory of aging that is being collectively disproved by the community right now. But if you want to write something about aging, you have to do methylation right now. In our first preprint, we actually also used electronic medical records. This is a signal that looks in some ways similar to methylation because it's 0 and 1 if you have or don't have a disease. And we were able to show, and that's the power of statistical mechanics, that you see the same things in all signals. So we were able to see that the entropy grows both in electronic medical records and in DNA methylation. And the properties of the entropy production were the same. We also shown, for example, that if you use just blood cell counts in order to measure the resilience fluctuations and entropy from longitudinal blood measurements, you have these estimates of loss of resilience, maximum human lifespan, the same you are getting from DNA methylation. What we're trying to do in our work is always use different signals. I think people just focus too much on DNA methylation right now. This will pass soon.
Yeah, it's just because it's the trendy tool.
Yes, but it's also in some ways more reliable than transcriptomics, for example, because transcriptomics has lots of distortion. That's very hard to combat. So that's why I think people were fortunate with methylation, because methylation is more robust. So that's why we have more papers about methylation. But what I'm stressing all the time is that when you think about phenomenology, you are thinking about universal truth that does not depend even if steam engine is consisting of atoms or not, right? So you're trying to work with variables that are universal. So that's why what you are describing is universal. You should be able to get it from medical records, from blood samples, from transcriptomics, from proteomics. So, for example, when we work with pharma companies and we use the models that we're training on electronic medical records, we are bundling them together with proteomics. That is now existing for hundreds of thousands of people, by the way. With this homologic and Olink technology, we are having now probably approaching 1 million samples there. Pharma companies are doing proteomics. This is a neglected signal because it's not in the public domain. You need to apply and things like that, so you cannot just download it and students do not fit linear regressions there. But this is a very powerful signal, because once you see something in plasma proteomics, then you get your target immediately, right? So with methylation, with genetics, you have to think a lot. What kind of gene products this gene variant is modifying, which is the tissue, lots of things to connect the dots. If you find your targets in circulating proteomics, you can do drug discovery from the next second on. So that's the answer to your question. First, we always try to prove that what we see in different signals. So that's why we have papers on blood work, we have papers on electronic medical records, we have papers on DNA methylation. We are happy to see that the results with DNA methylation are better accepted by the community. So everyone, we're building consensus at least on one signal. But we are making sure that what we see everywhere. And as a way to practical drug discovery, at least in 2024, my personal choice is plasma proteomics. That's the shortest way to do drug.
This relates to something else you described about in physics earlier, which is the separation between the sort of the people that are just collecting data, so they're just trying to describe the universe, and then the theoreticians. And I think in biology, I don't think we're doing enough massive data collection and also not making it public. So there's the Human Genome Project, which was a big thing. You know, like physics does this stuff all the time. Like they build the Hubble Space Telescope, they do CERN, and this data is public, right? And so physicists can work on it. In biology, this doesn't happen as much, which I don't really understand why. It seems like it's something that biology could benefit from learning from the way physics as a discipline works, is to have some portion of public funding going towards purely data generation, and then have the other portion going towards the theoreticians. You guys are not the ones that are generating the data right now. You're working on the data sets that can be accessed or purchased access to. But I would love to see, for example, the NIH spend 40% of its budget purely on data generation that the theoreticians could work on. I guess, do you think that,
I can reflect.
Do you agree with me?
Yeah, first of all, I agree with you. And second, I can reflect that on this. To make it work, why it works in physics, you need to have this phenomenology. So, for example, you have universe expansion rate measured by two methods that probably disagree a little bit, right? So you have phenomenology, and you know that this number should be the same from two kinds of measurements. And if it's not the same, people are going to funders, and funders fund experiments to bring these two numbers together or to show new physics, right? So here, I think why it's easier a little bit to convince funding, because people know exactly what they expect. And if there is any discrepancy, it's very easy to convince funders that this is the most important thing. As you probably mentioned from news, from physics, people live in this nightmare scenario when everything is explained, almost. And people are always looking for new physics. People have very hard time to show funders that, look, this will be surprising. It almost never works, unfortunately. But this also helps funders to focus on certain areas where the chance of progress is the most desired and hopefully expected. Without this kind of theory, where any result of experiment is possible, it's hard to know what is important and what is not, if you understand what I'm saying. You really need to bring your field to the situation where different people can agree what is wrong and what has to be measured. So you would ask different experts in physics, and many of them would agree that once this kind of discrepancy arises, many of them would agree that this deserves to be funded. In the world where any result of experiment is possible, it's just fun. I mean, why not those guys do that thing? You cannot decide.
It would be harder to get a consensus as to what data to collect. But we have things like, one model is the intervention testing program, where people actually vote on what things to test. And so perhaps biology could do a similar thing where we actually vote on what is the data that we think would be the most useful, and then the NIH just pays to generate it. Maybe that could be a different model. I think in biology too, there's a history of like empirical first, theory afterwards. Like Darwin himself traveled around the world just documenting species, and that's how he came up with his theory of evolution, was through just empirical data collection and then trying to understand the data afterwards. So I think in biology, but yeah, maybe that's enough said on that topic. But I just like to really always push for the fact that I think large public data sets would be beneficial to the field. And it was also in the Bottlenecks to Progress study that we started as a result of LBF0. That was the number one requested thing by people in the field, was more large public data sets.
I can quickly reflect on this as well. So first, I see that people in biology are very responsive. I think that, for example, for our last preprint, we got data from Vadim Gladishev's lab, which was very kind of them, but also we were struggling to find a good single-cell methylation dataset, for example. And we wrote to people from Elta's lab, and they helped us before the manuscript was published. So I think these things happen all the time, and I'm very grateful to these people. We have access to UK Biobank with a lot of data, so this is possible. So I agree with you that maybe some centralized effort on behalf of NIH may help, where these interactions could be automated somehow. I think there is a big value in what you are saying. And maybe just 2 words that, of course, I'm a theoretician. I want to build theory of aging, and I want to extend human life by a lot. But we're not only theoreticians. We're working with biologists, and we're testing our predictions experimentally. So when I told you, for example, about the proteome, we identified targets that are related to aging in humans, and hopefully we tested that in mice, and now we produced mRNA vaccine that is actually hitting that target, and hopefully we will bring it to humans. So still, I value good theory a lot, as you can measure from my words, and that's why I'm discussing it a lot. Theories are not worth much. They have to be tested, and for us, a test for a theory is a therapeutics eventually.
Yeah, so I just want to give a quick shout out to a project called the Systems Aging Institute, which is potentially something that could be funded by the NIH, that would be the purpose of which would be to pull all of the data together and generate more data. It could be a nexus, a hub for providing all the data that we need to build models of aging. And so if anyone in the audience wants to learn more about that, feel free to ping us and we can tell you about it. It's a project that I think, if it was funded, would be very important and could advance the field. But now, actually, you brought up something which I think is close to Nathan's heart. So I want to switch to the devil's advocate side of things, where we start challenging your guys' ideas. And I think for Nathan, one of the things that he worries about a lot is, how do we, when it comes to humans, how will we actually know if something is working or not, right? Like, you can test this in a mouse because you can actually do a lifespan study. But maybe, Nathan, you can elaborate on this, because I know this is like a major concern of yours. How would you even know if what you're doing is working in a human?
Yeah, so I think in our previous conversations, we discussed this quite at length at LB. When it comes to mice, we can do lifespan studies. So we can know whether an intervention extends maximum lifespan, median lifespan. We can measure it all. In humans, I guess the vibe in the general position in longevity is that we're never going to do like a full lifespan trial. You can measure rates in maybe all-cause mortality in maybe 5, maybe 10-year window, but there's no real plans to go any further. And then the other model that people have proposed for determining whether an intervention works is to do something like a tame-like trial, essentially measure the latency between comorbidities of aging. So the time it takes for you to develop diseases of aging, so potentially delay them. Last time we spoke, you mentioned that like the whole point of Gero is to find a large effect size. So how would we know that we see something, first of all, and how sure do you think, like just relatively in terms of confidence, that if that first signal, let's say that initial signal, will actually translate to maximum lifespan in extension in humans down the road when that's observed?
Decades go by.
Yeah.
Yeah. First of all, I think that's where a good theory is required, better with consensus and better challenged by different people. A good phenomenological theory should tell you that there are parameters like A, B, C, D, which are not lifespan, which together can be combined into lifespan, maybe in different ways in different species, by the way. But they should be present in all species. So that's why I'm talking about aging phenomenology, right? They just could contribute to lifespan in different ways, mice and dogs and humans. So it should be important to establish retrospectively that these variables, consensus variables of the standard model of aging, are related to maximum human lifespan from the data, at least. That should be a good guiding point. And once again, none of them should be lifespan, right? Because lifespan is a complicated measure. It depends on probability to get a disease, chance to get a good medical care, for example, right? A lot of luck. Lifespan is a bad measure, even though everyone is focused on lifespan in humans, especially, has lots of things that are not related to aging at all. So that's why we have to clean up our data, get rid of lifespan as a finite measure. I'm getting from your question that think about lifespan study, not necessarily at all. And then we should agree that there are certain quantities that are at least retrospectively related to maximum lifespan in humans that could be changed instantly. For example, the experiments that we're running right now is that since we have the standard model of aging, we know that there are certain quantities. We put up a preprint that, for example, mice also have entropic aging, which means that if you like, you can study drugs that affect entropic aging in mice. They wouldn't affect lifespan much, but they would affect entropic aging. Also, there are dogs, for example, from the data that we haven't yet published it, but it looks like dogs have extended entropic, extended part of aging that looks like human aging. And that's still much shorter than lifespan of humans. Still, we can measure the same quantities in mice, in dogs, retrospectively in humans. We can see if drugs that work in mice, in worms, against these quantities would also work against the same quantities in humans, for example. This is not the same as to run lifespan study, but at least you would see that the biology of in the standard model of aging is the same across the species. So my point here is this, that first we need to have this standard model of aging. We have to agree that there are certain quantities that are not lifespan, but they are important for maximum lifespan in humans. This has to be challenged, right? For example, I like this Calica effort I always cited. So when people started telling apparently them that there are animals that do not age, they funded a project to measure if naked mole rats age demographically or not. I think if community agrees on this standard model of aging, people will start measuring these quantities, challenging them from their data set. So eventually we will have confidence that these are the targets for interventions. And then once we start getting confidence that certain drugs, certain genetic perturbations, mRNA vaccines, doesn't matter, affect these quantities, we will have a chance that if everything is right, it will reduce aging. And then we should be able to detect, because the entropic aging occurs all the time, so we can measure it with DNA methylation, with everything else. We can measure them in mice. If we find a drug that affects entropic aging on the time scale of lifespan of mice, there will be a decent chance that we can put it into humans and see the effect. To me, if you don't have the theory, you are blind, you are just measuring lifespan, you don't understand anything. You get the best drug that works in mice, you put it into humans, you see a small effect, then you stop treating, the effect disappears, and you are... So that's my take on these things. We need to have the model, we have to start building consensus on the model, we need to have people challenging this consensus, first from retrospective data and eventually from prospective data.
Yeah, I like that interpretation a lot. It's having a standard of model of aging is like a benchmark too that you can look at whether you're making progress or not. There was an article, I forget the author, I'd have to look it up, from the semiconductor industry where he was talking about how we have benchmarks for performance and in multiple different types of them. And that's why you can actually have progress in improving computational power efficiency, different things like that. We don't have those benchmarks in biology. But I actually wanted to let Aleksandr maybe talk a little bit about this, because he had a really popular Twitter post recently that got almost 500,000 views discussing mice and the problems with mice models. And it's interesting to me because what you're talking about, Peter, is maybe it would be nice. I don't know if they keep samples from all of the ITP runs that they did, but could you go back in time and actually look at those samples and measure whether the trials affected entropic versus dynamic aging? And the same thing for the robust rejuvenation project. Like, those are potential sources if they have
the best
gained samples.
Yeah, this is probably one of the best experiments right now to look at. As I said, I'm sorry, Alexander, I'll just get back to you, because that's exactly where I think you should jump in, because you are working with the human data from the start. My point is that once you agree on this aging phenomenology, on the bunch of things that you should learn how to measure other than lifespan, you can even serendipitously find that a certain intervention actually reduces entropy, right? You don't need to know how it's been done, right? It may happen. that there may be an intervention that is otherwise overlooked, but is added maybe as a combination into Aubrey-de experiment that is just by chance reduces entropy as well, and that may be a great starting point for the future experiments, even though the effect on lifespan may be small. So that's why I agree with you that we should be collecting much more data on at least successful interventions, and we should be able to release them in such a way that people can analyze them. And large experiments like Aubrey's experiment is, I think, a great way to find by chance.
Yeah. I wanted to give Aleksandr a chance to comment on
Yes.
that too.
Yeah, just a quick side note regarding the ITP. I believe that for the first, like when they do a trial for the drug for the first time, they only look for lifespan. They don't collect any additional information about mice, and they only look for extra metrics when they do a second and third experiment. So since most of the stuff didn't work in ITP, they probably don't have
They
any
didn't collect samples. Could be a thing that we could actually reach out to them and suggest that they should be collecting samples even on the first round, because we could be missing important information.
That's probably the question of budget, right?
It's also the question of the budget for the RMR right now, which I think if somebody listening, or perhaps Gero, I don't know how much money you guys have, if somebody could pay them to collect samples, which could then be...
Right now, Gero is helping an experiment in dogs. That is a large longitudinal experiment in dogs. So they have stored samples. The experiment is over, but the samples are there. So we are helping to get denamethylation from this longitudinal bunch of measurements. So with some help, with some luck, we may know the answer by the end of the year if your dogs are more like humans, for example, in many subtle ways.
Any listeners with funding that they are looking to allocate towards an important project, this could be something to reach out to Peter or the NIH or Aubrey on.
Yes.
I think we could be getting more data from these projects than we currently are. Okay.
So I guess it doesn't really matter whether the data is coming from lifespan trials of any sort of model organism, as long as it's longitudinal and over several years or something like that. Then you should be able to detect the signal if there's an intervention that works in reducing the entropic production of aging damage or whatever. Yeah. And then how about in humans? What efforts can we do to find potential things that are intervening in entropy part of aging in humans, like existing data sets?
Yeah, I guess I can comment on that and what we are doing here. So at Gero, we are not only building a theory of aging and creating therapeutics, we also do a lot of experiments with different aging clocks. And some of these clocks are purely internal, but for some of this we see a potential. And one of these clocks is based on data from wearable devices. And you might be skeptical about it, but when you think about it, data from wearable devices is probably the most abundant data about human health we can collect, because like each smartphone collects your steps, like history of your steps. Your smartwatches collect your heartbeat, your HRV. Some of them collect oxidation levels and stuff like that. So we decided like we need to harness this data, which sits readily available. And we realized that there are several additional advantages which you can get by using the wearable data. First of all, this data is free to get. If user agrees to provide you the data, it's free.
Reminds me of SETI at home. You can just install the app on your phone and then you're just contributing to research, basically.
Yes, exactly. So this data is free. Then there is a history of steps, of heart rate measurements, of different kind of measurements done by wearable devices, all the way back to the time when you got your first device. So, for example, in my case, I got my first iPhone 5 or 6 years ago, and I have history of my steps since then. I can look back in time for this data. Same thing for heart rate. Additionally, since you can get access to this data remotely, you can pretty much start onboarding users from all geographic locations, and everybody has a smartphone. There is a very low barrier for entry. Plus, data from wearable devices are also easy to provide. It's just a click of the button. Like, you don't need to do any invasive procedures, no blood sampling, no troubles with doing saliva, nothing. You just click the button and data is shared. We were thinking on how we can harness this data, and we built an aging clock, which is the second generation aging clock based on steps. And the second model is based on steps and heart rate. And we built a simple demo app called Gero, which kind of showcases how it works. And one additional thing which I forgot to mention, since it's a continuous monitoring, you can continuously update your predictions. And potentially, you can start monitoring for interventions, because there is very high adherence from users. Like, data is collected without them paying attention.
Yeah,
without them
automated data collection is better than self-reporting, that's for
completely
sure.
automated. Exactly. And we built this demo app called Gero. Initially, it was just biological age calculation without anything. We provided it for free for the end users to play with it. But now we are thinking that we can start enriching this data by users providing extra context to us. For example, we can start asking users about their diseases, about their lifestyle choices, about their supplements, about their medications, about all of the diet, sports, fasting experience, and stuff like that. And we believe that we can find strong interventions there. We believe that we don't see any other way to find some of these factors. For example, probably one of the most important factor on your lifespan is your social status and your mental health. And it's really hard to study that in mice. We better start studying it in humans. And yeah, we're doing a simple platform right now. It's just a set of questionnaires for people in the app. It's a periodic questionnaire, and we can remind people with push notifications to complete it every 2 weeks, every month. Right now, we are starting with simple things. We are starting with mental health and lifestyle choices. We believe that even in this data, we can see something interesting because, as I mentioned, we believe that social status and mental health is crucial for your lifespan. And we actually see it in data when we experiment with psychedelics in model animals. That's why we personally interested in testing that. And right now we're just testing this approach. We see how many users will engage. It's completely free to join. Given that we collect enough data, we aim to publicly release it so that anybody can benefit from it and challenge us, publishing our findings for free. And in the future, we want to start doing other experiments. We definitely want to experiment with observational studies of microdosing because, as I mentioned, of psychedelics, we see the effect in animals. We want to partially recreate the study done by Matt Kyberline and other authors on off-label use of rapamycin when they did a survey-based study of different side effects of rapamycin usage. We want to recreate that. And if it goes well, we plan to create an open platform where people can create their own studies and vote for studies which other people proposed, and we can use our app as a platform for that. Yeah, that's
I
kind
just want to,
what that's probably the easiest and cheapest way to start collecting insights from humans. Definitely, it's not the most rigorous one, but we might find some low-hanging fruit there.
yeah. So it's a starting point, and I think it's also just like free and easy. So I definitely recommend to the audience to check out the GeroSense app. There's no harm in installing it, and it provides data that
Yeah,
will be
you
made
can read
available to
more on it.
researchers.
Yeah, you can read
But
more on it on our website,
yeah,
studies.gerosense.ai.
Yeah, but I do want to move on because the LBF in particular is not focused on longevity as a big umbrella. And there's the aspect of just buying time by optimizing lifestyle and things like that. But ultimately, you're still going to age and die. And the LBF is particularly interested in the technology paths to solve aging entirely. And so I think we should shift the conversation to that. And I think there's three areas that we probably want to talk about. But the three areas are the damage repair type stuff that Aubrey suggests, which I would call bioengineering, replacement, and cryo. And so those are biostasis. And so I'd like to get into your guys' thoughts on those, and maybe I'll start with the damage repair aspect and then let Nathan handle the replacement and cryo. And this is where I want to challenge you guys on approach. Personally, I think that we should be trying everything that's plausible. So it's not to say that your approach is something that I don't think should be tried, but it's more about there are other proposed approaches. So I think in the terminology that you use, Peter, Aubrey's approach would be to actually look at those forms of entropic aging and develop new mechanisms to actually repair them, right? So it's to make a list of everything that changes with age and then add new homeostatic pathways or treatments, therapies that could close those loops, right? Because we have homeostatic mechanisms for some things, but not for others. Humans have more than mice do. And in principle, you could start closing more of those loops and just be like, let's identify all these different types of damage that are accumulating and either look at other species that have ways of dealing with them and transfer that to humans, or invent entirely new mechanisms of fixing that type of damage. And that to me, that seems promising. There certainly seems to be certain types of damage that we could obviously just add another enzyme if we had a way of doing gene therapy, or and then that type of damage would just no longer accumulate. So I guess, what do you think of the damage repair approach, and what do you see as or are you skeptical? It seems like you're skeptical of it, but maybe I'd just get your thoughts on it. I guess, like, I would say that humans are already at the cutting edge in terms of our ability to repair damage, and so that might be a reason for skepticism, that improving on what humans can already do is hard. But there are organisms that live longer than us too.
Well, first of all, we enter into the realm of projections, which is always shakier than the realm of things that you already see in the data, or you try it and you know how it's going to work. My intuition is this. I mentioned it already before, that this idea, with all due respect, I think the obvious idea is a little bit reductionist, because it appears like you can list all sorts of damage. By listing, I mean that you can timely detect them, nicely localized. This thing is broken here, that thing is broken here. And then you're going to deliver many strong and timely and exactly delivered therapeutics that are doing the right things, possibly arresting the function of the cell at the same time, because sometimes you need to take out, I don't know, maybe a mutation that is now totally covered in chromatin and is not used anymore, on the region of DNA that is not used anymore. Technically, as I said, like in physics, that's what Boltzmann suffered for. His critics were saying that you can undo everything, right? Why not just turn the time arrow back, just change the initial conditions of all molecules in due time at sufficient accuracy, and you can go and you can move back in time. The problem is this requires high accuracy, high precision, and massive intervention against many degrees of freedom in your system. So intuition is that in such complex systems, when you touch one thing, you start touching other things. We don't develop interventions that only do one thing. All interventions do other things as well. And in theoretical mechanics, these things are called billiards, right? Because you change the angle a little bit and you are missing, and you change the picture a lot. So I think the intuition that we're getting from theory of complex systems is that if damage is done, and if damage is not correlated, right? If by changing one hallmark of aging, you're not touching all the others like you do in mice, then it's very hard to undo. which means that you should be focusing on things that prevent the future damage rather than undoing the existing damage. Because to undo the existing damage, you may end up having to develop infinite number of therapeutics, and we are not even near to that. So that's why prevention here works better than repair. And instead, you should be looking at features of damage-inducing mechanisms that are universal across all mechanisms, and such mechanisms exist. For example, the more noise you have in the system, the more damage you have. That's what doctors were telling us when we were young, that if you go to bed one time and wake up one time, you are going to get healthy. The less noise you have in your life, I'm not selling this as an anti-aging therapeutics, but what I'm trying to tell you is that prevention is better than repair. I think many people in our field underestimate the complexity, the immense scale of the task of repairing uncorrelated damage in the system with so many degrees of freedom. My guess is that within 10 years, if you ask me what will happen, what will work in 10 years from now, I think it's very reasonable to believe that somebody will develop a therapeutics that will reduce the rate of damage accumulation. And this will be far more likelier than somebody would develop many therapeutics and learn how to apply them precisely in order to undo damage at the same efficacy.
Yeah, I think when it comes to damage repair, you might be able to make a ranked list of perhaps which ones are more important, although that would be difficult to do. But for instance, oxidized cholesterol seems to be one that has like cascading effects, and Oki O'Connor's company is developing a repair mechanism to remove it. Prevention is obviously, if you can prevent, that's desirable. And I think your case is that maybe that's easier. I
Yes,
personally would like to see both be done in parallel, because even if you can slow the
sure.
rate, you still haven't stopped it. And if your goal is solving aging entirely, you will ultimately have to be able to repair this damage. Slowing the rate buys us more time to figure out how to repair it.
Yes,
So
that's my
I think we
point.
should
That,
I think we should do both in parallel.
yes, look,
Um,
as you're saying, make the rank list of things that are bad and try to develop a drug against each. So we have a rough budget,
Mhm.
okay? We have 10 things, 10 drugs, $10 billion, 10 years. Although 3 of them will be produced for sure. And this will buy probably 3 times 3 years, right? So we know that by reducing one major age-related disease, we'll increase lifespan by a few years, 2 to 3 years.
Yep.
And at the same
Yep.
time, please spare 1 billion for a drug that is reducing common factors that are driving different components of the noise. And you will have, if you develop such a drug, you will have a better impact for money. That's my line of thinking. I'm not against developing many drugs with precision medicine to be applied eventually to help. These could be, by the way, not drugs, but cellular technologies. For example, in our body, we have damage-removing systems, the immune system. I know that macrophages are superb in removing all sorts of damage. If you can clear up them a little bit, or if you can activate them a little bit, believe me, they will remove lots of things, much more than 10
Yeah.
things that you are talking about. So there could be very interesting solutions to repair. Our body knows how to repair many things. I think that's what people can do, will do. That's what biologists can do. This is the reductionism. This is on the top level reductionism
Yeah.
approach. I think we should go ahead.
Well,
We should complement
that
it
just
with something that is
yeah.
more universal.
Yeah, I think if you're thinking within the pharmacological paradigm, I think your way of thinking makes sense. I'm thinking more about the gene and cell, engineered cells and gene therapies as the approach, the new modalities that are currently rather immature, but as they mature and become more powerful, could potentially address damage repair. And so, to what degree is it that maybe you just think the timelines for developing more powerful gene and cell engineered cell therapies are long? Is that part of your thinking? Because yeah,
Well, let me,
I would agree
yeah.
that like pharmaceuticals probably can't do much about damage repair.
First of all, once again, it's projections, so it's hard to know. We know that since maybe last 30 years, since Genentech, people are always saying that small molecule drugs should disappear. We have antibodies, now we have vaccines, now we have gene therapies. So everyone believes that small molecules is some strange thing that should die. But they don't. Because as we, I think, increasingly understand, small molecules can do interesting things that gene therapies cannot do. Small molecules are quite promiscuous. They have many targets. much more than one target. It's actually exceptionally successful drugs have targets within a certain target space. That makes them very powerful. By hitting multiple targets, they sometimes can produce very large effect. It's very hard to evade such drugs because if you have many targets, mutations in cancer cells or any other mutations cannot lead to evasion. So my point is this: first, don't underestimate pharmacology. Great pharmacology does things that are not entirely understood right now. Also, in complex systems, acting against a single gene doesn't produce strong results. We know that we are products of sexual reproduction. We have at the same time many genes changed together. And still we produce a robust phenotype. We are human beings with different face shapes, but still we are humans. We still age at the same rate and everything else. So I think that point interventions against single genes may have limited power in aging. So I would not confront these two approaches against each other. There will be surprises. I agree that we should be looking more at cell therapies because, as I brought up this example with macrophages are Aubrey de Grey machines. They have a very long list of things that are bad for your body, and they eliminate infinite efficiency. It's just the problem that they also suffer from aging and lose their abilities. So I think we will see both pharmacological interventions that reactivate macrophages, for example, and we see limited evidence even in our experiments that this may work. I think people will produce engineered macrophages in the future to remove multiple forms of damage. It's hard to predict the future. What I'm trying to tell you is that even macrophages cannot reduce entropy by a lot. They can reduce a lot of damage, but there is a lot of damage that is beyond their level of recognition. And these things should be, I can actually elaborate. There are certain things in the work right now, but all of them that I see are still reducing the rate rather than removing damage. It's much easier from the things that I see or I can touch. It's much easier to reduce the rate damage production rather than to eliminate a lot of damage.
Yeah, I think within the synthetic biology community, there are some actually pretty interesting projects now. I think Seth Shipman's lab, Chan Zuckerberg Initiative are both funding engineered macrophages with enhanced capabilities. And that's still in the very, but they're also talking about adding new capabilities. But that's,
that would be super. Yeah, that would be.
Yeah. Okay. So I think we've talked about damage repair quite a bit. And I think the other alternative approaches, maybe I'll hand it over to Nathan to talk about replacement and cryostasis, because I think these are also, we'd like to get your thoughts on these approaches.
Yeah, for sure. Maybe we'll talk about replacement first. So I think because of the Nature Communication paper you guys wrote a few years back, you, Gero, and Peter, you guys became famous for saying that reversing aging wasn't possible. And obviously some of the people in the reprogramming field and so forth didn't agree. But Nature has figured out, in some sense, a way of overcoming entropy, not really a complete solution, but some sort of sidestep through reproduction. And essentially, reprogramming leverages this sort of hack, let's say this cheating. I think you've referred to it as cheating, restoring from a backup. But then there's other ways that you could do reprogramming in the sense of cloning, maybe not in vivo reprogramming, but a type of cloning to reset the state of a cell back to zero. You could clone a copy of your body. It would be genetically identical. Some people are working on trying to create non-sentient clones by genetically editing these cells so that they never develop consciousness. And then you would do some sort of head transplant. And perhaps that would solve aging in a, maybe it's elegant, maybe it's ghastly and crude sort of way, right? And maybe this could take longer than 10 years and maybe that's not on your horizon. But I'm curious what you think about this in general. Is this like a solution or is this more sci-fi in your view?
Yeah, look, so first if somebody would prove that aging can be reversed in humans, I would be happy. I think our field deserves a fight, right? And if everybody just agrees with everyone without producing strong effects, this is not the right way to do. So I'm prepared to be wrong, but I like to generate this discussion. So this is first. Second, with this epigenetic reprogramming, yes, nature found a way. So remember, I always talk about the second law of thermodynamics that tells you that if a system is sufficiently complex, it's hard to revert certain processes. So what nature is doing is that it's essentially killing the whole organism and produces just one cell out of it, which is immensely less complex than the whole organism. So it reduces complexity to pass this bottleneck, after which the rejuvenation becomes possible, right? So what I'm telling is that somewhere on the way between one cell and me, there is this horizon, there is a rejuvenation horizon set up by second law of thermodynamics. And there is only one way to pass it. And if you apply epigenetic rejuvenation to me, even if you do it the best way, like no one of my cells is going to die, which happens a lot during current, let's say, applications of rejuvenation. But I think this is technical, by the way, so I don't believe that this is a big problem. What you will have lots of embryonal cells. You will have 1 trillion me's. You will have 1 trillion embryonic cells, which is pretty bad, right? This is my, let's say, duty argument for the nature knows how to solve this problem. Nature first reduces complexity and then solves this problem, which is not the same as to solve the problem in its initial complexity. So second, I do believe that replacement is the most drastic or the most, I would say, brave way to counter second order of thermodynamics, right? So what you're telling to me is this, that, okay, let me comment on that. So maybe we should write at some point something about this. Because look, first of all, you can just produce these stem cells and shoot them into an old body. If our theory that aging is the compound effect of all damage in your body, then in order to rejuvenate your body, the intervention should be massive, which means that I should substitute at the same time a lot of pieces, right? Because if I just shoot one cell, the cell will adjust to the current version of the biological age that is dictated by your body. And I think there is evidence for that. You know that stem cells from old animals, if inserted into young animals, are functional. If young stem cells are shot into old animals, they are much less functional. So the environment matters. So once again, in order to make this idea work, the substitution should be massive. You should not be substituting one little piece by another little piece. Okay, where is the limit? So one cell is probably not much, right? People were injecting stem cells in older individuals, and the effects are not like 200 years of life, right? So something is not working there. Okay, you are saying that let's try to substitute an organ, or maybe to grow an organ embryonically, things like that. I think here we have now a balance. As I said, in order to work, such intervention should be massive, which means that you have to produce a massive injury. Think how much, I don't know if there are measurements to that, but I assume that if you do a massive surgery, or if people have firearms wound, for example, right, an equivalent of massive surgery, I bet their biological age is going over the roof at that point, right? In order to do the intervention you are suggesting, you have to have such precise surgery that doesn't have any other effects on your body. I think that may require certain technologies that may not yet exist. We know that if you do any massive surgery, it may lead to sepsis, and you can kill a person with massive surgery, right? So my argument here is this. So if I were given money and we're raising funds to do what we think we need to do, I think that how to do whatever, genetic intervention, vaccine, drug that would reduce the rate of aging, this is within reach. We only need to find the right target. This is target not like a molecular target, the phenotype, which is responsible for the rate of aging. This could be done by a community of people working on that within 10 years. If you need to grow organs, this is possible. And then look how long people live, how people with organ transplants are living now. And these people are not exceptionally healthy. So I think in order to achieve, once again, first it may work, I agree with you, but to achieve it technologically, I think you have to build a stack of technologies that do not necessarily exist or cannot be made be applied on a mass scale. This is my intuition. I know that, at least in physics, when people stopped shooting themselves after realizing this entropic problem, people learned that trying to go against the entropic increase is the last thing you want to do as an engineer. If there is a way around that, try to do that first. And maybe in the future, when people will start growing organs inside you in the right places, I don't know, maybe in the future. I just think that a mass product can be developed in 10 years that will halve rate of aging in humans with the technology that we have. That's my kind of, it's not like argument against.
Yeah, this is something we thought about it when we were building the roadmap, which is like, who is the roadmap for, right? Because if a plausible technology development path is like 20 to 30 years, then obviously certain people will never benefit from that. But at the same time, I think from a strategic perspective, it is, I think, important to pursue multiple plausible paths. And the advantage to the replacement path is, I think, that there's less unknown science. There is surgical technique. You're correct in that, but that's more of an engineering problem, not per se an open science question. And so the idea, and like you say, also do more at once. Do, I think, the whole body at once would be what we would think makes the most sense rather than an organ at a time, because every surgery, like you say, has risks. And so if you do the entire body at once, then by definition, that your whole body is now young if it was a clone of you. And cloning would be one of the reasons that people that get organ transplants now actually have shorter lifespan relative to somebody who didn't need an organ transplant is because they have to be on immunosuppressants. And so cloning, a cloned body would be the thing. But again, the timelines involved in that are long. The lead times are long. And so it does have disadvantages, but it also has the advantage of being maybe more of an engineering problem than a science problem. So that's why we think it's an important alternative strategy that should be pursued in parallel.
I can support what you are saying is that no matter what we think, this discussion is set up as to what should we do in order to dramatically improve human lifespan. But I think if we think about this cloning and organ replacement, far before this will work for increasing lifespan, it will help a lot of people who have acute organ deficiency problems, right? So I think this is a step-by-step development where you can benefit both financially and scientifically right away if you do it. There is a clear problem that needs to be solved. For example, the problem with organ failure cannot be solved pharmacologically. So there is no choice, right? You need to develop an organ transplant. You cannot develop a gene therapy that will sort these things out. So that's why I agree that this is some technology stack that has to be developed eventually to solve the problem, right? So maybe within 10 years, my take here is that within 10 years, if we put enough focus, because I've seen drugs that reduce phenotypes that I believe are responsible for the rate of aging. So these drugs exist. So it's not like a big science. The science is to prove that these phenotypes are responsible for the rate of aging, but that's a different thing. I think that if both technologies are developed, genetic rejuvenation will also help in a bunch of diseases, by the way, right? So I think realistically we will have a bunch of new drugs or organ transplant technologies, including cryostasis, by the way. Cryostasis will work, I think, first for the organ transplants, right? And then maybe eventually to recover people from death. I think it's just a wave of technologies that are being developed side by side. What I'm trying to develop will not solve any medical problem, so I'm at risk, right? Because I want to reduce the rate of aging, which is not a medical problem, which makes it in a way more complicated in terms of fundraising and investment and everything else. But all the other guys are doing decent medical stuff like organ transplant supporting technologies, and they will collect lower hanging fruits. So I think there are different moments to that, pros and contras, and all this technology should be developed.
Yeah, and I think an important theme that we discovered when we were doing this roadmap is how intertwined some of these technologies can be. Like they can be synergistic and applied in different ways, or might be necessary to help, develop one pathway to the other, like bioengineering might be useful in cryostasis for developing like cryoprotected proteins. And then the stuff that you guys are doing at Gero, if you can drastically slow down the rate of aging, then you wouldn't need as many, let's say, whole body transplants if that was like a solution that you wanted to do. Yeah, because even if you slow aging, you're still aging at some point. You'd have to figure out what to do with an aged body. And
people will still have diseases, by the way, right? Slowing aging doesn't mean that people will suddenly have no diseases. It's just that diseases would not be age dependent anymore. But still, there will be the whole spectrum of the same diseases. The same doctors will do the same things. The total number of people will increase by 50% still. So you'll still have to have a lot of organ transplants and replacements. So I think having all these things first discussed in a respectful way, like we do. Thank you very much. Second, being put on the same roadmap with discussion that these things have solve direct medical needs. These things solve more scientific interest. I think having them on the same roadmap and seeing this thing evolving over the, let's say, 5 years from now, will get objective measurements on the rates of these processes, right? Now you have just one point and intention. You have one point and direction. I think if we work hard enough and together as a community, and if you would follow, or let's say 2 years from now, 1 year from now, 2 years from now, on your roadmap, you will see the rates. And this would be very important because you would be possibly reporting that certain things are underfunded, underdeveloped, right, and important. And certain things are just not going anywhere because nobody could estimate the amount of difficulties that are there. So I think it's a very important starting point, but also it's no less important is to make sure that we're looking at it from a side and objectively measure, because nobody can predict the future of the progress.
Yeah, definitely. You mentioned cryostasis already, and Mark brought this up as well in the sense that our roadmap, when we started it, we asked ourselves, who is it for? And we wanted it to be for basically everyone. Like, no matter what age you are, there should be some sort of solution or pathway for any person at any age. And even if we could arrest or freeze the rate of aging so it doesn't increase exponentially, for older people who are in their 80s, 90s, maybe they're 100 years old, that mortality rate is still high. And even if it's just stuck there, not increasing, there's still going to be this exponential drop off in
the aging is not the biggest problem
population.
for these guys, for sure.
Yeah, exactly. Yeah, they just have a really high mortality risk. If there's no way to rejuvenate them in the near term, then it might stand to reason that a strategy would be to just employ another trick of physics, which is to slow down.
I think we could be, I'd say, once again, we're going into the realm of projections, but we know that the brain size of people in the advanced stage is pretty small, which means that people should be looking at things like Neuralink prosthetics, for example, just to regain the brain volume effectively by technology. And this goes pretty close, I think, to mind uploading and things like that could develop in totally unexpected ways, for example. So people may start losing identity by horizontal transfer of experiences and things like that. So once again, it's hard to predict how these things will end up. Most probably these things, I'd say, we're talking a lot about aging because we're confined in the physical body. And these things will also be disrupted eventually, also at a rate that we may not understand correctly. So I think maybe on that end, we should also talk more to people who are thinking about brain prosthetics, because brain is just one thing that is very hard to replace, by the way, right? Maybe adding those people to the community, to the roadmap, and maybe by interacting with them more, we may end up thinking about more interesting things, because non-aging is just one thing. It's just, to me, it's just a human enhancement, right? So, for example, when we see this age-related decline in IQ, I mean, no kidding about that happens well before any disease. So maybe we should be thinking about drugs that could try to arrest that. And it looks a lot like these drugs would also arrest the true aging, the entropic aging, right? So there should be very interesting overlaps if we try to refocus our thinking and our messaging to this kind of enhancement, physical enhancement, cognitive enhancement, arresting this age-related decline. Then suddenly I think we may be interested, we may get interesting to larger population of people, right? Not necessarily aging freaks, let's put it in a respectful way like this. And they also have resources, they also have communities. So I think we should start thinking that way as well, both pharmacological and technological.
Yeah, definitely. I think Neuralink and other brain-machine interface, brain-computer interface companies are on our roadmap for a slightly different reason, not necessarily for uploading, although I guess that's one potential solution. Yeah, we mostly included it in the sense that it could be a solution for head transplants. If you
prosthetics, yes. Exactly. I also started from that. I agree with you. So it has to be part of the narrative.
Yeah. But what do you think about cryostasis in general? Because it's the original slowing of aging by using physics.
Yes, but this is a very extreme. Yeah, this is one thing that I'm afraid of, because if you look at the reversible processes that occur throughout our lifespan, The formation of our memories is one of these kind of hard to reverse, fortunately, processes, which could be seen from outside, like another entropy production part. And of course, if you want to shut down this kind of irreversible processes, the remote risk, which I see, would be that if, for example, Gero will find a drug that would reduce whatever noise that is responsible for the rate of aging, the first system that would suffer from that would be the neural system, because neural system relies on noise for operations. So then we will possibly end up in this biostasis situation. We will have zero rate of aging, but no mental processes going on. So I think these things are related. To me, biostasis is still an extreme way of solving the problem. So it's still undesired way of solving problems. But as I said, it solves a medical problem, like organ transplant. It may solve the medical problem, so it has to develop. And then learning to operate at lower rates of noise, maybe for medical reasons first, and then eventually maybe for life extension. Why not? I look with a lot of respect to this field. I hope that they will progress.
Yeah, we're really thinking about the people that may not survive to see the development of the other technologies we're talking about. If there was a reversible full freezing, full pausing of aging, that would allow them to be revived when we can actually solve these problems. And cryo has been a chronically extremely underfunded field. I would say there's a lot of plausible hypotheses for how you could achieve full cryostasis reversibly. And there are even natural examples, right? There are actual animals that exist in nature that can be frozen and thawed, and they're perfectly fine. So we already know that it's not impossible. It seems like it's an engineering challenge. And so I think that's why Nathan and I definitely feel like it's an important part.
Yeah, I think here the organ replacement and biostasis are actually competing. These are the two things that, fortunately or unfortunately, competing as part of the immediate medical problem solution. And then this, unfortunately, I think may keep the biostasis funding at low limits for another long while, because I think with all this embryonic development of new organs, I think this thing is being funded right now. They show some interesting results. I think this field is eating biostasis.
Yeah. But what about preservation of your brain? Are either of you two signed up for cryopreservation?
I'm not signed yet, but I probably will as soon as I settle geographically, since for me it's pretty cheap these days. It's just a monthly insurance fee.
Better than being buried in the case that you happen to get in a...
Exactly.
I think we've covered the major sections of the roadmap.
I did want to get one hot take from Peter. You know, when I talked to Emil originally, before I knew anything about biostasis, he nerd sniped me by just saying the whole problem in cryostasis, it's really just a physics problem. We just need to really figure out how to rapidly rewarm a cryopreserved or vitrified specimen from minus 200 degrees Celsius. And that got me thinking. I was just like, oh, it's just a physics problem. My background is also in physics. And it set me down a little path to investigate further. And we've had other people in our fellowship say things like, oh, yeah, like also from engineering backgrounds or physics backgrounds say, oh, if it's just a physics problem, we just need to get a couple of smart engineers or physicists to tackle this problem of essentially heat transfer. So I'm curious if we can borrow your brain for, 30 seconds and anything off the cuff, if you've thought of any strategy that could rapidly rewarm a brain-sized vitrified object.
For the 30 seconds while you've been asking this question, my brain produced a joke that boiling an unboiling egg is just a physics problem. So why not? All of that is, look, I do think that we have this, especially engineers, have this idea of reversibility, right? Like we can do whatever we want in every direction if we have enough funding. I think we need to get a little bit humbler. So to me, make something that is alive to age a little bit slower is less ambitious than try to stop something to something that looks almost like being dead. And then it's just physics, right? Why not just boil it back to life? No, I think we underestimate the complexity of resynchronization of a complex body. I understand that animals do that. Most of those animals have proteins that not just substances like glycerol. Some of them have very sophisticated proteins that arrest crystallization of water in every cell in their body in multiple copies being expressed right before. So for them, it's not a sudden event. They are preparing their body for this situation in such a way that they can actually survive that. It may be that, once again, this is a fascinating area of studies. When I learned about those proteins, I was really impressed that there is the whole sequence of things that these animals are doing in order to survive this procedure. And we mostly don't have these mechanisms, and these mechanisms have to co-evolve with those proteins for a number of years, millions years. I'm kidding here. So once again, my current approach is, I think, so if you let me reflect back on our conversation, so at every branching point of this roadmap, I'm trying to choose the way that does not require a dramatic, whatever, rewiring, reset of the system. I'm trying to take gentler turns every now and then. Because I believe that whenever we are thinking about massive organ replacement, about recovery from frozen state, right, we are thinking about such a dramatic change in our body that the sequence of events should be perfect, much more perfect that I'm typically doing with the help of my biology friends in a typical biology experiment. So that sense of humbleness probably makes me take gentler turns at every step.
Yeah, I think that's a fair assessment. And a lot of the approaches that we look at the roadmap, we measure their difficulty level in multiples of the Apollo project, because that's like the unit of measurement that we think of. And it could be that many of these approaches are much more difficult than that in terms of the amount of science and engineering that would be necessary. And I agree. Yeah, these organisms have fairly complex mechanisms to facilitate being frozen, and I suspect that it might require us to do some fairly complex genetic modifications of humans in order to facilitate reversible cryostasis. That's my personal view.
Yes, and this has never been demonstrated to this level, even for simpler examples, I believe. So that makes this, to me, a little bit more remote than everything that we're doing. Because remember, also in biotechnology, the timescales are just ridiculous. If you have just a wonderful idea today and assume that you know that everything works, right? Your idea is perfect. I mean, when is going to get to humans? 6 years? 5 years?
At the same time, we haven't prioritized it. Like the biotech research is a haphazard, piecemeal, Brownian motion wandering. We're not doing big goal-directed large projects like the Apollo project. Like maybe if we decided to spend $250 billion to solve the gene delivery problem, maybe we could do it in 10 years. But that's not currently what's happening.
Unfortunately, in a free society, you need some crazy dictator doing that.
If you want to vote for me.
That's not how dictators.
You can do the job.
Cool.
I agree with you.
So some final thoughts. Who are the top people that you follow in the sort of field? Are there any people that you follow on Twitter or labs that you like to follow their publications? Who do you think is important for our listeners to be paying attention to in terms of getting good information and ideas? Other than your,
because don't follow me under no circumstances, because I'm writing some physics crap all the time.
It's hard to read, yeah.
It's hard to read, so don't read it. This is over. You've been warned. No, I always enjoy, for example, reading Matt Chamberlain. I like his no bullshit, almost Russian physics style approach and comments on biology projects. We need something like that. I'm subscribed to different labs that are not longevity, or laboratories that are not longevity laboratories, because I think biotech is much larger than aging. We should be getting ideas from that, and we should be interacting with that. We're just too small as a community. We have very ambitious goal, but otherwise we're small, which means that I definitely think that people should be described to some people who are popularizing science, from David H. to Sabina Haushofer, for example, popularizing physics. She's doing, I think, a very good job on bringing up physics news. I kind of a physicist, but I'm reading that a lot. But I think solving aging is a massive social engineering, like Manhattan Project or Apollo. It's not just an engineering problem. So that's why we need to tell somehow into the current, let's say, cultural narrative to make sure that what we are proposing is interesting to many people. So we need to read and listen what kind of problems biotech is now trying to solve, to convince them that solving aging is part of the solution of those problems. So that's why I'm trying to interact with those people. I was, for example, impressed over the last 2 years how much pharma is actually interested in aging, right? So they're interested, they're not impressed, but they're interested, they're reading everything that we're writing, not us, but as a community. I think we can find very interesting readers among them if we actually achieve good results, but also if we frame our narratives in such a way that we will help solving their problems. I think this will be also very helpful. So that's why I'm reading mostly people who are doing modern drug discovery, and I'm trying to see how aging may be a part of that.
Yeah, for me, I also highly recommend Matt Kyberline. Yeah, he's usually doing a great science and also insightful information. I also recommend, maybe surprisingly, Peter Attia, who is quite pessimistic regarding all longevity field and our potential to extend the lifespan. However, I think he's doing a great job in kind of summarizing what we know to date for humans in practice. I definitely recommend subscribing to Tomorrow Biostasis on YouTube. They post great educational content, which very honest in my opinion. Yeah, that's all for me.
All right. And then our final question is, how can our listeners help you guys out? What do you need? Are you hiring? Are you fundraising? What could help you guys move faster? What are your bottlenecks to progress?
We're looking for people with exceptional abilities, as usual. So we're looking for data scientists more than biologists right now. But let's say we're open. If we see an interesting person, we will try to interact. Obviously, we're always fundraising, but who doesn't? But money also help. We are at the stage where expensive experiments are required. More and more expensive experiments are required, and money will soon be a bottleneck. Not now. Now we still have a few scientific questions that could be resolved faster with better funding. But I think in a year or two, money will be the bottleneck. So interesting people who want to solve aging in big way, and people, adventurous people who may help to fund and help develop a company that would solve aging in big way. Everyone is welcome because it has to be getting faster. We should accelerate.
Agreed.
Yeah, and everybody with a smartphone can join Geoscience studies.
Yes, and join the Gero study because there will be some surprises out of that.
Great. Looking forward to following your guys' progress. Any final thoughts, Nathan?
No, it was a great conversation, and I want to thank Peter and Aleksandr for coming today, as usual. And yeah, very excited. I find just always inspired, maybe because I'm very biased with a physics background myself, by the way that Peter thinks. And I think it's just like such a unique and needed sort of perspective to the field. And yeah, I'm looking forward to
Thank
seeing
you for
all the progress.
for having us
Gero.
every time.
Thank you.
All right.
It's our pleasure.
Have a good
Thank you.
time, guys. We'll see you around.